Hello, and welcome to my poster presentation about BrainTrawler, a web-based framework for the exploration of heterogeneous spatial big brain data. My name is Florian Ganglberger, and I'm a postdoctoral researcher in the Biomedical Image Informatics group at the BioVis Research Center in Vienna. In recent years, big brain initiatives created vast resources of spatial brain data that can be used by researchers for their own projects. Understanding the relationship between genes, structure, and behavior is one of the driving questions in neurocircuit research. This requires the fusion of spatial brain data of different modalities, such as whole brain gene expression, structural and functional connectivities, or non-spatial data like gene lists related to behavior or functional associations. There are typically problems with this. It involves often a very time-consuming manual aggregation of the data from different resources. Those resources are not necessarily on the same reference space, don't have the same resolution or the same anatomical level. Therefore, we created BrainTrawler that allows a joint exploration and fusion of this data from brain network data on a voxel or a regional level, which can have billions of edges, or spatial gene expression data with 10,000s of images. A workflow in BrainTrawler looks typically like this. You start with a 3D and 2D visualization of your reference space, here in this case the mouse brain. You select then a region of interest by either brushing here your region, or you select a brain region from an hierarchical ontology. Then you select the connectivity, the network data that you're interested in, like structural connectivity, and hit target query or source query. This gives you the aggregated outgoing for the target query or incoming for the source query connectivity from this selected brain region. It is indicated here on a voxel level in purple and on a regional level as this node link diagram that you can see here. Quantitative information is here shown in this bar chart, which shows you the mean connectivity from this region in the anatomical regions that are indicated here by the same coloring. This matrix, the raw data of this matrix, has 100 gigabytes, and this query can be done in real time. We just have to wait for 1 second. To achieve this, we created a data structure for real-time aggregation queries. It basically uses spatial indexing to make these queries much more efficient. So you basically reorder your connectivity matrix, which is a representation of your network, where every row or column is a voxel. So on this row, you see the outgoing connectivity of this voxel. So you order this column so that voxels that are also spatially close in space are spatially close in this matrix, perform a row compression and store it on your hard drive. This means that the connectivity data of your brushed region here is also close here on the hard drive and can therefore be read much more efficiently instead of loading the whole matrix in your RAM. We also implemented some caching mechanisms here, and you can also retrieve this data due to a hierarchical ontology on different levels. But for this, you can look at the paper which is referenced on the bottom of this poster. So what you can do, so we did here for an example 3 case studies. The first one is you started with genes of interest, visualized this just as a volume, looked at the overlap, which is here in this brain region called the central amygdala area. And from this area, we performed target queries for structural connectivity here. And we see that it has a strong connectivity to this area, bed nuclei of the strata terminalis, which is known from literature. We also have here the second connectivity, which is also high in this area. And when you look at the overlap of both, you see that this connectivity from the literature is confirmed. When you retrieve this on a regional level, you can do something very nice. You can navigate to the brain regions you are interested and find, for example, correspondences here in the mouse brain and in the human brain. So since these are two different ontologies, we have different coloring, but neuroscientists selected here brain regions that correspond, and we confirmed that we find an overlap in specific connectivities across species. What we also have here is a genetic dissection of connectivity. So we selected here this brain region of interest and performed a so-called gene expression query. So we took for 20,000 genes the volumetric images of gene expression and put that in a spatial index, similar to this as we had for the aggregation queries. So we queried very quickly and efficient the genes that have a high mean expression in this area and retrieved a list. We also did this on the targets of this area. So this was a target query. We selected the boxes that have a high connectivity, performed also here an expression query. And we can see now that from those two lists, you can select, we want to have genes with a high expression here and a low expression here. And the genes here represent then molecular mechanism. So thanks for your attention. Please have also a look at my short presentation about BrainTrawler, which is much more about our ongoing projects. And if you have any questions, please just contact me via this email.